started on the 12th. Today we'll be starting with Ezenwa from University of Georgia.
So first I just want to thank Dana, who's out there, for the invitation to this meeting. It's been sort of fun to listen to the discussion yesterday and the talk yesterday. And so I thought sort of today I would start with this idea of host heterogeneity. That's something I'm really interested about. It sort of nicely follows up on some of what Felicia talked about, host quality. And sort of one thing that I see as an important sort of feature that disease ecologists care a lot about is this idea of host heterogeneity, or essentially sort of variation that we commonly see in hosts in sort of their ability to get infected with something, as well as how likely they are to infect others. And so there's sort of a broad array of evidence that a lot of intrinsic host traits, so things like age, sex, your genetic background, and behavior can influence those things. And sort of one thought is that, you know, a lot of what we know, or, you know, we think we know maybe about some of the mechanisms that might link things like sex, for example, to, you know, being more likely to spread infection or being more likely to acquire infection, might come down to physiological and immunological differences of the host. And so this might be a reason to sort of, for disease ecologists, to try to open that box, the black box within the host, and sort of eco-immunology slash immunology and what's going on inside of the host. And so while there's been a lot of attention on these intrinsic host traits, one thing that sort of gained more recent attention, and a lot of people in this room work on these questions as well, is this idea of co-infection. So maybe it's not just things about the host itself or things about the environment, but maybe other parasites and pathogens that can drive this heterogeneity or variation in a host's ability to acquire or spread infection. And so the exciting thing about co-infection is that it's ubiquitous, right? So we know that a majority of hosts are actually infected with more than one thing. And I just like to show this graph, which is in people. And so if people are co-infected with so many things, like think about all those natural populations out there. But this is just co-infection in people of different ages in a village in Ivory Coast. And it only looks at helminths in protozoa, so just two types of potential parasites that can infect this taxonomic group. But we see that really early on, so, you know, by the age of 1 or 2, a lot of individuals are infected with multiple parasites. And then there's people that, you know, the few that are infected with up to 10 different species of parasites. So given this sort of wide spread and common feature that a lot of individuals have all these other parasites, one interesting point that arises is what effect do these other parasites have potentially on your ability to acquire or spread sort of incoming parasites. And so there's been sort of a large body of work sort of at the within host level on these types of questions. So ranging from looking at parasite interactions that might be between microparasites and microparasites, so things like viruses, bacteria, and protozoa. And so we know a lot about sort of how immunity, so a lot of focus of these within host parasite interaction studies have been immune function based, and so we know that immunity can drive things like, you know, interactions between herpes viruses and bacterial infections. A lot of work looking at sort of macroparasites and microparasites. So now you have parasites that aren't necessarily that similar, yet they still, they're potentially immune mechanisms that can cause them to interact. And then more recently, a ton of interesting new work even broadening that idea, saying, well, maybe a lot of host-associated microbes, whether they're pathogens or not, are interacting potentially and influencing the ability of a host to acquire and spread or clear infection. And so a lot of different types of interactions can happen between this sort of broad community of host-associated microbes, whether they're pathogenic or not, within the host. And so what role does that play? And if sort of disease ecologists are interested in understanding sort of broader scale patterns of disease, so be it, invasion of disease or broad scale patterns of prevalence or spatial distribution, so where is the pathogen found and where is it not? In addition to sort of thinking about all of these extrinsic factors, maybe we should consider things that are happening within the host and sort of look at them side by side. So one thing that I'm particularly interested in is sort of the second thing here, the macro-micro interactions. And there's some reasons why they're sort of an interesting, potentially tractable set of questions to study. So first start in the macroparasites, sort of I'm focused on in particular helminths. And I just like to show this picture because helminths are some of my favorite parasite group. But they're fascinating in a number of ways. They're sort of gory, but they also sort of infect almost every organ of the vertebrate body. So they do a good job at what they do. And we know that sort of from human studies alone that sort of the parasite or helminth burden of humans across the globe is huge. And so we know that there's these sort of widespread distribution of these parasites. One thing that sort of interested me early on was a lot of conversations with vets who said, well, they're so widespread that maybe they don't have any effects. Which seems like, I think this is having an effect on the host. But either way, so there's sort of this broad group that are so widespread and endemic in a lot of natural populations. So one question really is, so aside from sort of the direct effects they might be having on the host, what are the indirect effects they might be having through things like their effects on other parasites and pathogens in the system? So in particular, I got interested in this and sort of looking at gastrointestinal nematodes in particular in African ungulates in sites like this. And this is just a sort of a snapshot of 11 different species of ungulates at one of my study sites and the prevalence of infection at any given time of individuals in all of these species. So the majority of individuals in all of these different species are infected with these nematodes and simultaneously infected with other types of parasites and pathogens. So what role, if any, do these pervasive sort of nematode infections have on how we sort of understand the interactions between the host and these other parasites that are often sort of less prevalent and sort of non-endemic. So one thing that makes sort of studying helminth microparasite interactions interesting is sort of what we know or what has been established in basic immunology about how these parasites influence the host's immune system. And so just to sort of boil it down into a cartoon, and luckily lots of people like to draw nice cartoons about the immune system, which is one thing I like about immunology, is that, so basically helminths can influence the host immune system in two main ways. Well, in many ways, but if we boil it down to sort of basic paradigm, so one way is sort of via stimulating this Th2 type 2 response. And so sort of the paradigm is that when you upregulate this response, then it comes with this cost of potentially downregulating Th1 response, which might be directed towards intracellular type things. Simultaneously, you can get these parasites also sort of stimulating or mimicking the sort of regulatory pathways of the host as well. So in addition to this sort of cross-regulation, you might get sort of general immunosuppression as well. So these parasites can sort of not only bias the host sort of in one direction over the other, they can also just cause general immunosuppression. So in combination, helminths might have the potential to sort of strongly influence other types of pathogens that might, that a host might sort of come into contact with. And this has been sort of the basis of a lot of work in laboratory models as well as in humans, this idea that sort of helminth effects on the host immune system might have a strong effect on how hosts respond to diseases, or in the case of humans, things like HIV or tuberculosis or malaria. So a lot of interesting work with interesting patterns at that scale. But one thing that sort of has become more in interest, I would say, to some ecologists is, well, can we understand how those within-host processes, which in this case we might think are the purview of ecoimmunologists or immunologists, well, can that help us predict sort of between-host consequences? So something that you might traditionally think was the purview of disease ecologists. And so this seems to me as one important area of interface where we can really use that information about immunology to understand some fundamental things about how diseases are distributed at any level, right? So it could be sort of some of the classical things that disease ecologists might be interested, like invasion or the probability that some microparasite might emerge, persistence, so patterns of prevalence across populations. It can even be things like parasite species richness. Well, how is it distributed across hosts? And does co-infection have anything to do with that? As well as the spatial distribution of parasites. So potentially, one question is, well, what role does immunology and understanding these within-host processes potentially have with helping us sort of understand large-scale patterns of disease? And so if we sort of think about, so stepping back to think about a sort of basic sort of disease population level ideas, and so following nicely up on Steve, who talked about the classical Anderson and May stuff, is just sort of in a cartoon form to say, okay, if disease ecologists sort of view microparasite diseases in this sense, right, as sort of classical compartment model where you divide up individuals as being sort of susceptible to infection, infected, recovered, and then we sort of look at the transition rates between these compartments to say something about the potential for a parasite to invade a population maybe, or try to understand its prevalence. And then we just say, oh, let's introduce the helminth parasite, right? So what if the helminth parasite, this host population is infected with that? So how does this pre-existing infection potentially influence some of these parameters that might help us say something about disease distributions at a population level? And so sort of going off of these basic ideas about how helminths affect immunology, and then empirical laboratory studies that have sort of shown some patterns out there, well, we could say that basically, you know, if we expect helminths to down-regulate or suppress the host immune system toward these microparasites, then these parasites might change these different parameters that we're interested in, but I'll talk about this first. And so if we want to focus mainly on this idea of invasion maybe, so isolate one variable in terms of disease that we're interested in, we could think of sort of this idea of R naught that came up yesterday as well. So the basic reproductive number of a pathogen. And basically sort of R naught is a function of sort of parasite transmission. So transmission has to be sort of higher than the parasite loss from the system. And so these parasites get lost through mortality, and it could be disease-induced mortality here. It can be natural mortality of the host, or the host could just recover from infection. And so basically when transmission exceeds that loss or sort of the duration of infection, then R naught is greater than 1 and the parasite is likely to invade. And so if we care about microparasite invasion, we can ask this simple question, well, how might helminth co-infection affect these different variables? And so if a helminth is suppressing the host immune system, then potentially, right, you release the microparasite to replicate more potentially, and so it's more likely to establish in a host, so the host becomes more susceptible. If the host is immune suppressed and the parasite is replicating more, you might increase the host infectiousness, so any given host's ability to infect another host, and so in combination those things increase the transmission rate potentially. So on the other hand, right, if the microparasite is replicating sort of much faster because of this immune suppression, the disease might progress much faster in that host, so you enhance the likelihood that that host actually dies. So you might increase disease-induced mortality. And then again, of course, if sort of the host immune system is immunosuppressed, you might lengthen the time to recovery, right? So you might reduce the probability that that host can clear the infection. And so these are all the things that a helminth infection could potentially do if we just use sort of translate sort of basic ideas from immunology into these sort of key parameters that have been established in disease ecology for looking at sort of population level patterns of disease. And so one interesting thing here is that helminth co-infection might, change any one of these, or it might change them all simultaneously. So really sort of looking at this in this framework might help us understand, well, where there is variability, how can we use that to understand perhaps when co-infection effects that happen within a host might actually have a consequence at some sort of broader scale, so it might have an ecological consequence. And so the one interesting thing that also arises from looking at this in this framework is this idea that in general, helminth co-infection might increase the transmission rate. So, hosts are more susceptible, hosts may be more infectious. So you uniformly perhaps increase the numerator of this R naught equation, but the interesting thing here is that for the denominator, two things can happen. You can sort of increase this, so you can increase the denominator here by having a lowered recovery rate, right? So, so helminth infection might reduce the recovery rate. So basically, by reducing the recovery rate, you lengthen the infectious period. So the parasite stays around longer in different individuals. But by contrast, you might actually reduce the length of the infectious period if these hosts die faster. And so you get two things that can happen in the denominator, and that introduces potential variability in how we expect co-infection to actually play out at a higher level. And so one question that sort of follows from that is, can we use this sort of basic, simple conceptual idea to understand when co-infection should actually have these ecological consequences. And are there sort of attributes of either the pathogen, the host, or the environment that we can use to make predictions about cases when we might expect to see these strong population level effects. So here focused on this idea of invasion versus not. And so you might think, well, anything about the pathogen maybe that alters how it influences sort of transmission relative to the infectious period might be a variable that we could consider that could change the outcome of co-infection. Similarly with things like season. So, you know, different seasonal factors that might influence how the host invests in its immune response could change the outcome for sort of the transmission box versus the infectious period box. And so I just wanted to discuss some basic ideas about how characteristics of the pathogen might help us think about when we expect consequences and when we don't. And so this is work in collaboration with Anna Jollis. And sort of first, we've looked at this idea of persistence and severity. So can we use ideas about how long the disease persists? So basically, is it an acute infection that has this sort of short time frame, or is it a chronic infection that persists for a long time? And also, how severe is the disease? So is it highly lethal? So fairly severe, is it fairly mild, where most hosts actually recover? And so the interesting thing about these two things is that they sort of have variable outcomes when we think about them relative to the infectious period. So like I said before, you know, in any case, we might expect sort of transmission to uniformly increase. So if a host is immunosuppressed, perhaps one of two things always happen. It gets more susceptible to infection, or it's more infectious. And if this is happening across hosts, maybe sort of transmission rates across all of these pathogen types are sort of uniformly increased. But what about the infectious period? So if we think of sort of an acute pathogen that's severe, well, if it's severe, then in fact a helminth infection by suppressing host immunity might allow for faster disease progression, and that might enhance the disease-induced mortality, so the host might die faster. On the other hand, it might also reduce the clearance rate, so it takes hosts longer to recover from infection. And so using a basic sort of additive idea here, ignoring the magnitude of these effects for the moment, we might just expect that those effects could actually cancel one another out. So you have no infectious period effect, right? So everything is driven by transmission, and the net effect on R0 is some, you know, average level of increase due to this pathogen. But then if we think about a mild infection, right, so there's no disease-induced mortality because most hosts recover. And so in fact, there's no sort of effect on alpha in this case. But on the other hand, the disease is persisting longer in different individuals. It's because of this reduced clearance rate. So that increases the infectious period overall, and so now we get sort of a combination effect where the helminth is increasing transmission and also potentially increasing the infectious period. So we might predict that for acute mild infections, we'd actually see stronger sort of population level effects, at least on this sort of R0 value or initial invasion of a pathogen, than we would for something that was acute and severe. And so if we use these same ideas for chronic infections, and so the idea with chronic infections is that sort of hosts never recover. So everything is driven by mortality. because it persists for a long time, you're infected for life. So it comes down to mortality. And so if the infection is mild, then there's actually no effect. And everything, again, is driven by transmission. But if the effect is severe, and then potentially the infectious period effects can counterbalance the transmission effect. So for things that are chronic and severe, we might see the lowest sort of effect in terms of invasion potential compared to things that are acute and mild. So perhaps we might expect that strong effects of helminths are more likely in some types of pathogens than others. In this case, things that are mild and acute might sort of be the most likely to be associated where we have these population level effects associated with co-infection versus chronic and severe things. And so one interesting thing is sort of in the literature, particularly on humans, there's a lot of focus on the chronic and severe things because indeed those are the things that we care about a lot. So things like TB, but indeed the population level effects of helminths on TB might be very difficult to detect even though we see them at the individual level. And so this, of course, as I mentioned before, this is sort of a basic way of thinking about these ideas, but it does provide some insight. And one thing that is important is sort of what's the relative magnitude of these effects, because that ultimately will determine the true outcome. And also there's the issue of sort of dynamical feedbacks as well. So, right, helminth co-infections are ultimately influencing the population dynamics of the pathogen and vice versa. So sort of understanding these things in sort of a theoretical context is perhaps an important next step. But anyway, so using these sort of basic ideas, we just went to the literature and asked, is there any evidence for this? So is there any evidence that if we try to pull out variables that have been measured in these laboratory studies on mice and sort of slot them into these parameters that you could fit into a disease model, is there any sort of signal that you do tend to see if you calculate the net effects in the same way? Stronger effects on certain pathogens than others. And indeed, it seems to be that sort of the clearest effects, and so these studies just cover the last 10 years of co-infection work in laboratory models. But if you look at these studies, then it appears that sort of mild and acute things tends to cluster at the top here as predicted. And so perhaps these ideas have some predictive power. And the mild chronic things tend to be the things down here where sort of there's equivocal support, I guess, for what the net outcome might be. And so again, these might be sort of ways to explore, well, what do we really expect when we think about co-infection and when we think about the likelihood for co-infection effects to scale up. And I think a really interesting way to sort of combine ecoimmunology and disease ecology, because one thing is that sort of if you go to immunological studies, it can be hard to sort of put metrics that have been measured in those studies into sort of a disease parameter context, and sort of vice versa, without thinking about sort of some basic immunological ideas, sort of organizing this information can be difficult. So that seems like one sort of nice intersection between the two fields where sort of if disease ecologists considered immunology, they might sort of start adding some of these kind of things to their models and how they think about disease distribution, and vice versa. Immunologists who are doing all of these really nice studies in the lab might take measurements that sort of help us apply them directly to some parameters of interest in studying disease at the population level. And so one thing that we are interested in our own work is sort of trying to understand the magnitude of these effects then and what those really mean. So as I said, that sort of conceptual model ignores the idea that there's the magnitude of these effects is important. So some preliminary work we've done in African buffalo, where essentially we treated some animals over the short term for their helminth infections, and then measured what the effect of that treatment was on sort of an immunological parameter, and sort of used that as an estimate of the potential transmission effect. And so in TB, they don't recover. So then the infectious period is completely determined by mortality effects. And so then we also use sort of parameters estimated by what the combined effect of these two infections was on condition to sort of estimate what that mortality effect might be. And then looked at, well, what is the effect of variation in nematode transmission on the basic reproductive number of TB. And so sort of these results suggest that these effects are driven by the transmission effect. The co-infected mortality effect in this case has a little effect, and so the combination sort of suggests that nematodes can indeed influence the R0 of TB. But again, these sort of parameters are estimated, and we're now in the process of trying to get at what they really are in a long-term study. So the second and last thing I wanted to talk about was this idea of site of infection. And so the other thing, sort of pathogen biology trait that might influence and help us predict what we might expect is where the pathogens are actually located within the host. And some people like Andrea and Amy have done some interesting work discussing ideas like this as well. But so one idea is that, so if you have this sort of cross-regulation or Th1, Th2 trade-off idea, well, it's really between intracellular parasites and those extracellular helminths. And so you have an effect here on intracellular parasites, but these helminths are also inducing sort of general immunosuppression, and that can affect both intracellular and extracellular macroparasites. So we might expect perhaps that we'd end up with a stronger magnitude of an effect on intracellular microparasites than extracellular microparasites. So just generating a simple prediction that we might be able to go out and test. So we've been interested in this idea of, okay, well, so if we see these types of population level effects, do those actually then scale up even more and determine patterns of microparasite richness across species? And so do these cross-population effects actually explain some cross-species patterns that we also see? So in collaboration with Charlie Nunn, we've been doing work sort of looking at patterns of parasite species richness across primates. So using a database, the Global Mammal Parasite Database, that includes records of parasites and pathogens from wild primates that were collated from the literature. And so essentially we took all this parasite information and classified the microparasites according to whether they're sort of obligate extracellular pathogens or obligate intracellular pathogens. And we wanted to ask this question, so do we see sort of any signal of a difference in the magnitude of an effect? So the basic prediction was, right, that if at a population level, helminth infections and the prevalence of helminths are somehow facilitating microparasite invasion, then maybe across species, we expect that species that have accumulated more helminths and have higher helminth prevalence might accumulate more sort of microparasites. But this effect might be stronger for the intracellular ones because this combined effect of immunosuppression might be greater than for the extracellular microparasites. And so sort of initial tests of this idea have shown, and so basically the analyses here have used the Bayesian MCMC analysis to take into account phylogenetic signals. It's something that's really important for these cross-species analyses. And the other thing considered here is sampling effort, which is a big issue with these analyses, is that, you know, the number of studies on particular species strongly determines the number of parasites that have been discovered in these species. So sort of using 90% support of this as this cutoff for sort of strong support for a pattern between the variable of interest, so here would be sampling effort, here would be prevalence, intracellular and extracellular microparasite richness. We see that, so there's 100% support, right, that sampling effort is a predictor of parasite richness, and yes, because the number of studies strongly correlates to the number of parasites that have been found. But the interesting thing we also see is that there's 91% support for the idea that Hellman's prevalence is strongly correlated to intracellular parasite richness. And then we see this strong drop-off in support for those extracellular microparasites. And so this suggests in these simple bivariate tests that there perhaps is this difference. So one, Hellman's prevalence might indeed be correlated to variation in sort of the establishment of intracellular parasites in across primate species, but there's much less support for them driving and influencing extracellular parasite richness. So the second question, and one thing that sort of came up yesterday, is this idea of, so, you know, how important necessarily might these effects be? So the Hellman sort of perhaps immune-driven effects versus other effects. So in sort of a number of studies that have looked at what are the sort of broad predictors of parasite species richness in mammals, so spanning primates, ungulates, and carnivores, well, three things have emerged: that life history traits like body mass, behavioral traits like group size, and then sort of just characteristics of the species like their geographic range size might strongly influence the, do strongly correlate with parasite species richness. So in rerunning these models sort of in a multivariate context and looking at, well, what's the support for the role of Hellman prevalence versus the role of some of these key life history traits. So again, we see this idea, so of course sampling effort has very strong support, but so interestingly, Hellman prevalence has as strong support as body mass, which has to be the variable that's most strongly in every analysis that's almost ever been done correlated with parasite species richness in mammals. We see that basically there's sort of equivalent levels of support as with geographic range size. So this suggests sort of preliminarily that Hellman prevalence might sort of have effects that are somewhat similar to some of these key life history metrics, so intrinsic host properties that people typically measure and study in this context. So some final thoughts on integrating ecoimmunology and disease ecology is sort of this idea of co-infection and disease control. I think that some of the things that arise from that conceptual model is this idea that if sort of population level outcomes are driven by the infectious period, then it's sort of interesting to note that we can't always use sort of the individual level impacts to make predictions about what should be happening at the population level. So if you take really severe diseases that cause mortality, so mortality is a huge and severe individual level impact, but it's perhaps moderating the population level effects. And so when we control disease, we might just consider those larger outcomes. So just one thing that sort of, if we think about control only in the context of individual level effects, there might be counterintuitive population level outcomes that we aren't considering. So perhaps just a place where the combination of these two fields can add to the how things are considered. So already sort of disease ecology and ecoimmunology provide a lot of ideas for things like vaccine coverage and development, but this might be sort of drug therapy might be another place we might think about this, since there's increasing calls for using antihelminthics, right, to treat a lot of, you know, high impact, severe human infections. So perhaps thinking about it and incorporating disease ecology might be useful. And then this idea that even understanding within host effects and population level outcomes. So this is my plug for wildlife models, which people in this room have talked about. Amy Peterson had a great paper on wild immunology. So just this idea that to really understand this, we might not ever be able to use lab models to do it, because if we need to do population level experiments or long-term studies of populations to understand how helminths actually drive disease over time and space, it might be difficult to do that in laboratory models, and it's really difficult to do it in humans. But thinking about wildlife models might be a way we could really do this. And to do this, we need a lot of ecoimmunology, which comes back to points discussed yesterday, to even start figuring out how to measure some of the things we have to measure in these potential wild model species. So that's all I had.
Plenty of time for questions.
Yeah.
Yeah, Vanessa, it seems like there'd be a real potential for helminth infection to drive down beta in the behavior sense. If you think about human helminth infections like onchocerciasis or filariasis or hookworm, they really increase morbidity. And so if transmission of microparasites is dependent on individuals being out and about,
Yeah, moving around,
and they're not going to do that because they feel so bad, or they're actually handicapped by, you know, lymphatic problems, you maybe would see beta drop dramatically.
Yeah, so that's a great idea that sort of, it's not, yeah, sort of these immunological things are important, but also behavioral things. But then you think about sickness behaviors, right, are also linked to the immune system. So
Right.
maybe the idea that they're staying home and they're lethargic. Also, we still have to understand sort of immune basis.
Right.
But
so
that's a great.
And I mean, those systems in particular seem like they would be really amenable to testing that easily.
Yeah.
Because we've got such big data sets of people, of the epidemiology of those helminth infections and the associated epidemiology of pathogens.
Yeah, and sort of the behavior of the people, like things at work and stuff like that. That's a great idea.
That would depend. It would be different for a vector transmitted disease versus a directly transmitted disease. The vector transmitted disease might not care that you're immobile.
That's true, unless you don't contact the vector, like outside your house, you know, if you never go outside.
That's
But
right.
yeah, that's
so
interesting.
a lot of vector-transmitted pathogens, the vector behavior is what really matters.
Yeah.
Some vectors are totally unwilling to go inside houses, so it's going to be highly dependent on the vector of the pathogen of interest. But you're right.
Yeah.
I thought the result of the intracellular parasites and the helminths from the primate database is really interesting, and I'm wondering, I'm sure you thought about this, and I'm just wondering what your speculation might be as to, might there be reasons why exposure to intracellular parasites and helminths would be positively correlated, such that it's not the co-infection that's driving it?
It's a correlated exposure.
Right.
Yeah, so that was sort of the rationale for looking at these different types of microparasites, right? So if you see this strong correlation with one type, sort of intracellular microparasites, well, there could be all of these other things going on. So trying to sort of say, well, do we see different effects that follow from sort of some potential a priori predictions for two different types of microparasites? So it appears that we see this very strong support for one and not for the other. And there's not that I could think of yet, sort of a priori reason why helminths might increase exposure to one stick but not the other. Although, I mean, but I guess it doesn't follow necessarily with intra versus extracellular, where a lot of microparasites, for example, are transmitted in the same way as these helminths, but they cut like fecal oral or something like that, but they cut across both classes. Yeah.
So some microparasites subvert the immune system by provoking a Th2 response when a Th1 response might be the most appropriate one, like RSV or Mycobacterium leprae. So if you had a parasitic infection, that would be predicted to sort of push the immune system toward the Th2 type of response. So RSV or Mycobacterium leprae would be now a really lethal disease instead of just a bad disease.
Yeah, that is interesting that there are these exceptions that I think are really interesting. So I sort of presented this sort of case based on some major paradigms, but there are some really interesting cases like that.
The detail that would be.
Yeah, where you would expect for some things sort of even more severity than other things. So sort of trying to get that magnitude idea. But also cases so that where if microparasites actually, depending on how they cause the severity, so if it's sort of immunopathology related, you can sort of sometimes get alleviation by having
I think
the helmet. So
I think these two cases, they are highly infectious.
yeah,
And actually, you suggested that the helminths were increasing the risk of infection from the microparasites, which is presumably a bidirectional response, right? So I'm wondering if you have any information from your studies where you maybe removed microparasites so you can get an idea of whether the strength of the direction is, whether the direction of the magnitude is stronger in one direction versus another.
yeah, so that's a great question. That presumably this is a bidirectional relationship, and so yeah, we don't have any studies where we remove the microparasite, which is sort of an idea that for logistical reasons failed, because it's much more difficult to do that than to do the reverse. But yeah, I think that there are definitely cases sort of in our study system, for example, where you get very early vertically transmitted microparasites. And so sort of looking at this idea of, okay, well, what are microparasites doing to the macroparasites as well, I think is an important question. So all of those different interactions that I talked about, so like, what are microparasites doing to microparasites? Macro, 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 micro in opposing directions. I think they're all interesting. So we've just sort of been using this macro, micro as sort of one case study or paradigm to think about these issues. But I think thinking through them for all of these different combinations of types of parasites could be really interesting.
If it is stronger in one direction than another, then the order should make a huge difference. Right?
Yes, so the order does presumably make a huge difference, and that's sort of one reason why we focused on this order, because these sort of helminth infections in our system come early and stay forever, basically. But then there are these cases of some vertically transmitted microparasite things that are even earlier. So that's sort of an interesting case in our study systems, for example.